In the second part, we will focus more on the kinetics of DNA repair using the gamma H2AX assay that we have already described previously. So if you remember, we were now at this point, and we already saw that essentially that thanks to the phosphorylation of the H2AX histone, which is one of the histone that make the chromatin where the DNA is bound, and thanks to the fact that ATM phosphorylates gamma H2AX for about 1 megabase pair around the position of the double-stranded break, we can actually visualize very nicely double-stranded breaks. These pictures that you see here, these color pictures, these are all in stained samples. So you have to fix the samples, you have to dehydrate the samples. You take the sample on a slide, you put the sample on a slide, they are dry, they are dead, of course, and then you do this immunostaining with different antibody, as we have seen before, a primary antibody and the secondary antibody, and then you look. You also use another stain generic for the DNA, like DAPI. We have seen when it is blue, it's called DAPI. This is propylium iodine, which is red, and then you look at the fluorescent microscope, where essentially you see these fluorochromes. You excite the fluorochromes with a lamp in the UV region, and you see the emission of light in the visible spectrum. In this case, in the green spectrum. So that's how it looks like. You can do a lot of things. This is an example here of colocalization of foci of different proteins to sites of DNA double-stranded breaks. It's you see this is RNF 168, 53 BP1, MDC1. These are all proteins we will see involved in DNA double-stranded break repair. And of course, if you wait enough time that these proteins are actually recruited to the sites, you will see images with different colors where you see the colocalization of these proteins. That's already a way to know whether this protein is involved in DNA double-stranded break repair or not. If you see the focus in correspondence to gamma H2AX that you know is involved in DNA double-stranded break repair, then you conclude that it is involved. But the interesting thing is that you can also see the number of double-stranded breaks induced by radiation as a function of the dose, and the kinetics of repair. Kinetics of repair that we have already seen from all the experiments that generally is described with a double exponential, because there is a fast repair kinetics and then there is a slow repair kinetics. But you see here, this is actually done by counting. The gamma H2AX assay is simply done by counting the number of spots. Normally it's done at the microscope. You can also do automatically at the flow cytometer, so you look at the intensity of the signal, but it's actually more accurate if you do it counting, maybe with some kind of automatic counting system. You see that the efficacy of repair strongly change depending on the cell line. That these are different tumor cell lines here. Some of them are repair proficient, they repair very well. Essentially, there's nothing left after 20 hours. Some of them seem to be repair deficient because, as you see, after 20 hours, there is still a substantial residual of DNA damage. This is 100% the initial damage, and this is the residual damage. So you can look, you see, these are different cell lines, you have different residual damage at 24 hours. Now, Marcus Lobrich, this is again from the University of Darmstadt here, made a very important experiment a few years ago when he actually was able to use gamma H2AX to measure the induction of double-stranded breaks per cell from very, very low dose up to, you see, up to something like 1-2 gray. to measure very high dose with the gamma H2AX is impossible because, of course, you see everything, all the dots start to overlap and you cannot count them anymore. But these points here at very high dose, you see this is 10 gray, this is 100 gray, these are done with the old technique of pulsed Tholgil electrophoresis. We have seen it in the previous lecture. What is really amazing here is that going from 1 milligray to 100 gray, so we are spanning some, we are going through some 5 orders of magnitude. The dose response curve is remarkably linear. So it's really the response, the induction of double-stranded breaks per gray is remarkably linear over many orders of magnitude. And you see, at 1 gray, this experiment was done in fibroblasts. At 1 gray, the fibroblasts are the cells of the connective tissues. You see, at 1 gray, the number of double-stranded breaks is something between 20 and 30. 20 and 30 double-stranded breaks per gray per cell. What is also remarkable is that in this plot, you see it was possible to measure the dose in the milligray region. And then this begs the question, how is the dose repair when you go to these very, very, very low doses? You see here, this is 20 milligray. The black bars represent the background. Of course, you always have a background which is technical, so somehow you never get rid of some noise in the signal, or is also real, corresponds to real double-stranded breaks in the cell that are spontaneously occurring and then they are repaired. So if you give 20 milligray, this is the repair time in hours. You see, so you go from 5% of an hour to 24 hours. You see that at 20 milligray, you have a pretty good repair. You see, essentially, here you have a clear excess, which you can also see here. At 20 milligray, you are here, 20 milligray. You see, this is 10 milligray, so this is 20 milligray. So you have a clear increase compared to the background. When you go to 5 milligray, you still have repair, but you see, it looks like it's less sharp of what you have at 20 milligray. When you go to 1.2 milligray, you essentially have no repair. This experiment is very controversial, of course. It looks, if you remember, we said at the beginning that one factor affecting DNA repair is tolerance. So whether some damage can be tolerated by the cell without any action. And apparently here, you see, if you are in the order of 1 milligray, no repair is activated in the cell machine. So this is a bit controversial because later the same authors, Marcus Lobrich, they did an experiment in vivo, look at CT scans, which is also very low dose. And you see, this is now the foci as a function of here is not the dose, is a gray per centimeter because comes from the way you calculate the dose in a CT scan. So it looks like linear, but in this specific case, there was actually repair. You see, this is the control values for two kinds of patients: those that have a very large field. So this is almost a whole body CT, and instead in blue, in dark blue, you have patients with a small field. It's only chest, only CT of the chest. So you see that half an hour after the radiation, you have many more foci, many more H2AX in these patients than in these patients. But then apparently both of them are able to repair. You see, both these patients and these patients you see a reduction in the number of foci as a function of the so it's still not perfectly clear whether the milligray are completely ignored by the cells and not repaired. Now, these studies that we have seen so far with gamma H2AX are essentially snapshots. So you take a snapshot at a certain time. But if you want to see the real kinetics, if you want to see the recruitment of the cells and the repair, you have to try to work in live cells. How can you work in live cells? Actually, the best way to have fluorescent proteins in live cells is to use this famous protein, the green fluorescent protein. The green fluorescent protein, which is a typical beta barrel structure. If you remember the secondary structure of the protein, this is a beta barrel structure. This protein was actually isolated from a jellyfish, so a jellyfish which is fluorescent, by a group of Japanese researchers. They got the Nobel Prize in chemistry in 2008 because GFP really changed the biology a lot, because transformed the fluorescent microscopy that was traditionally done in fixed samples in live cell imaging, where you can actually use proteins that are labeled with this GFP. How can you use these so-called reporter genes, you know, these genes that have this marker, this fluorescent marker? Either you introduce the gene by transfection, you introduce this fluorescent genes in the cell by transfection, or you have in the genome, you have the gene itself, which is producing the GFP protein together with the protein that you want. In either case, you are able to see the fluorescent protein in the nucleus. Of course, if you look at the live cells under the fluorescent microscope without radiation, without any special action, you will see everything fluorescent because this protein will be a little bit everywhere. But if you have recruitment of the protein to sites of damage, there you will see a specific focus, just like you see, you know, here in gamma H2AX. You see, this is the recruitment. This is just like in gamma H2AX is not the recruitment, no, because gamma H2AX is a protein which is stable there, it's not moving. But if you have a GFP protein, which is actually like 53BP1, MBC1, RNF 168, these are all proteins that are moving in the nucleus to go to repair, then you should see the movement, you should see the recruitment in this fossa. And that's in fact exactly what you can see here. This is now 2 cells. The cells are U2S. This is a tumor cell. It's an osteosarcoma. It's called U2S. The protein that we are looking at in this cell is MBS1, the Nijmegen breakage syndrome 1. You remember one of the proteins involved in if you have the Nijmegen breakage syndrome, you are deficient in DNA double-strand break repair. And this is 53BP1. 53BP1 is one of the most important factors in the repair. You see also here, if you look at the time, you see starting from 0 here, this is the speed of recruitment of NBS1. And you see that the speed of recruitment of 53BP1 is lower. 53BP1 comes a little bit at the end. But you see that these movies are accelerated, of course. You see that eventually they are nicely seen on the screen. So here you can now play. I told you that we understand the mechanism of repair of double-stranded break. So we know which proteins come first and which protein come later. You see here, this is MRA11. This is MDC1. MRA11 is part of the MRN complex that you see here. MDC1 is here. RNF8 is here, RNF 168 is here, and 53BP1 is the final big focus that you see here. Is the complex that eventually involves the double-strand breaks and repair it. So you can see here the recruitment of all these different enzymes, the time of recruitment of all these different enzymes, and you can put them in line. You can see which enzyme is recruited first, who is recruited second, third, and so on and so forth. This is again an example. This is RNF 168 versus 53BP1. You see that the timing of recruitment of these two is similar, with 53BP1 coming slightly slower than RNF 168. And you see that after X-rays, of course, you see single dots. Imagine that every one of these dots is a double-stranded break, but this time you don't see the phosphorylated gamma H2AX. You see the recruitment of 53BP1 to the site of the DNA double-stranded break. And this is fast. You see, everything happens already in seconds. You have a strong recruitment, and then in minutes, you see this whole movie is 6 minutes. So it starts really in a few seconds. So it's surprisingly fast. You know, the recruitment of so many proteins to the sites of the damage is very fast. The processing is slow, so then it takes hours, you know, to fix. But the initial steps are extremely fast. Now, what happens if you have heavy ions? Of course, as you can imagine, you will have a tendency to have more complex damage because this is densely ionizing radiation. So if these are the tracks in nuclear emulsions going, you see this is increasing LET from protons to iron down here, so the LET is higher. This is how it looks like in the cells. This is a gamma H2AX. It's not a recruitment, it's a gamma H2AX assay. Gamma rays, you see single points, but when you go to silicon, silicon is here in the emulsion. This is how it looks like. It's wonderful if you want, it's fantastic. I mean, what the physicists were predicting, you know, that the damage was going to be along tracks because of the physics of the process is what actually happens in biology. The DNA damage now, because this is gamma H2AX, the DNA damage is along the tracks. And this is iron. You see 3 tracks of iron, and you clearly see 3 tracks of double-stranded breaks. You also see some delta rays here, some damage which is done off track by the delta rays. Of course, you can play with the colocalization. This time is even more funny because you see, this is with X-rays, 1 hour after irradiation, 8 hours after irradiation. This is MRA11, gamma H2AX, and this is the overlap of the two, showing that MRA11 is recruited with the gamma H2AX. But if you irradiate the cells, you know, not going inside the screen, but parallel to the screen, you see these beautiful tracks, you see, and then you can stain, for example, this is gamma H2AX, you can stain gamma H2AX, you can stain ATM, and you see, I have to put this one here, and you see that ATM is colocalized with gamma H2AX correctly, because ATM has to go to gamma H2AX and has to phosphorylate ATM go to H2AX and phosphorylate it to transform it in gamma H2AX. Here you have, this is the gamma H2AX in this nucleus. This is DNA PK. Again, they are nicely localized, you see. Gamma H2AX and MBS1. We already saw that MBS1 is an important protein in DNA repair, and in fact, they are localized. Gamma H2AX and CHK2. CHK2 is actually a protein, is a checkpoint inhibitor. So not necessarily is colocalized with the gamma H2AX, but surprisingly it is. So CHK2 is another protein which is actually goes to the site, is involved in the DNA repair and in blocking, if you want, the cell cycle progression, because it's recruited to the sites of DNA double-stranded breaks. And these are snapshots, of course. So there is no kinetics. Can we have kinetics? Of course this is another example. Again, this is RPA, 53BP1, and this is the overlap. This is not so obvious because keep in mind that RPA is actually involved in resection, while 53BP1 is involved in repair of double-stranded breaks. So if you see an overlap, and you see an overlap, not everywhere, but you see a lot of green and red, you see an overlap, this means that in this particular case, the cell is going into resection of the double-stranded break, which means alternative non-homologous end joining. This specific case is possible because this cell was irradiated with uranium ions, so the damage is so complex that it's resolved by using RPA. Now, can we do live cell imaging with heavy ions? Of course we can. Of course, in this case, you have to put the microscope. So this is Burkhard Jacob, who is the expert in our group for doing, this is a facility really only at GSI. The other images that you have seen with X-rays were done in the Netherlands, but this is done only at GSI, where you have the microscope on the beam line. This is the monitor of the camera. So when you shoot the beam, you can actually see the image of the cells live. And as an example, you see now this is these are human cells, U2OS, this osteosarcoma. The enzyme is aprataxatin, which is another in the complex of 53BP1 involved in DNA double-strand break repair. Uranium ions, and make no mistake, the beam is shot perpendicular to the plane, so it's entering in the screen. Okay, it's not going in this direction, let's say in the lateral direction, it's going into the screen. So when I click play, it's like turning the accelerator on, and you see that, and this is a live movie, it's no accelerator. You see that in a few seconds, you start to see the dots. Each one of these dots is one uranium ion. But make no mistake, it's not one double-stranded break. There will be many double-stranded break along the track, right? Many double-stranded break along the track. But you see only one, because of course it's coming in this direction. So you don't see the distribution of double-stranded breaks in the track. And again, look how fast it is. Of course, if you irradiate parallel, these are 1, 2, 3, 4 cells, you see now clearly this is MBS1 green fluorescence protein in U2S, and now you see many tracks now in the transversal direction going through the cells. This is nickel, 350 milliliter nucleon. This is actually 53BP1, U2OS, uranium 750 MeV per nucleon. 53BP1, I told you, is a little bit slower because comes at the end. And in fact, you see, it takes some more time to see the streaks of 53BP1 generated by uranium 750 MeV per nucleon. This is XRCC1. XRCC1 is interesting because XRCC1, the particle is iron, 1 GeV per nucleon. XRCC1 is a single-strand break repair protein, which means this movie is going in loop, which means that after a while, if you look carefully, the nice track that you see will disappear. Disappears because the protein goes, fix the single-stranded break, and then leaves, because repair of single-stranded breaks is relatively easy. So you see the track, but then the track faints away. In 3 or 4 minutes, it's already gone. And same here. This is now XRCC1 again, but this time with uranium. So you see many tracks. These are many cells, actually. This is a low magnification image. So you see many cells, and you see these beautiful tracks of XRCC1 formed after irradiation with uranium. And it takes some time to disappear in this case. You see more time, because of course with uranium, you create so many single-strand breaks that even XRCC1 stays for long, but you see in many cases disappears. I mean, in some cases you still have a strong signal. You see after this is already more than 10 minutes, 15 minutes are gone, because it's it's 1,000. It's 1,000 seconds, so it's 20 minutes or 30 minutes. But you see, now it's gone almost everywhere after 1,500 seconds. Now, one of the strange things that you can see sometimes are things like that. You see, looks like this is not a movie, of course. This is a snapshot. Sometimes looks like the track going through the nucleus is interrupted, you see? Or sometimes even worse, looks like it's turning around. These cells are mouse embryo fibroblasts. In these cells, it's very clear the difference between heterochromatin and euchromatin. Heterochromatin are these big blobs here. This is heterochromatin. In between the heterochromatin, this dark but almost empty spaces, this is euchromatin. So this is the area highly methylated, transcriptionally inactive of the heterochromatin. And this is the area hypomethylated, transcriptionally active of euchromatin. Now, first time I saw it, I mean, this is amazing. It's so strange. You know, how can a particle turn? This is, and you can also see here, looks like this is now a colocalization, you see, of gamma H2AX, RPA, that shows, you know, the turning around these blocks of heterochromatin that reminded very much this picture of Salvador Dalí in 1932. You know, this is the track, and now this is the heterochromatin, and looks like the particle is going around. Impossible. No, it's impossible. Because it's impossible because the particle has to go straight. So to understand this, we actually switched to live cell imaging, and we used the XRCC1. This is the famous XRCC1, the protein XRCC1 that you have seen before, specialized in single-strand break. And you see these red blobs are heterochromatin, and in the middle there is euchromatin. Now, if you look now at the kinetics, boom, you see, now you see and you see that these tracks actually go through the heterochromatin, just like through the euchromatin, but the repair is much slower. You see that the repair in the euchromatin is much faster than the repair in the heterochromatin. This you can see it also here. You see the track, now there are two cells here. You see the repair in the euchromatin, but you see how slow is the repair in the heterochromatin. Now, boom, there is another track coming. And again, you see that the repair is much faster in euchromatin than heterochromatin. So what happens? You can see this in a zoom here. This is the heterochromatin. Now, when the track comes, boom, and here it is. You see that it is repaired here, and it actually leaves the heterochromatin, goes out of the heterochromatic block to be repaired outside. That's why in the snapshot of the gamma H2AX looks like there is a turning. There is a turning because the damage is moving, which is actually produced inside the heterochromatin, but to be repaired has to be moved outside of the heterochromatic block. So this is, you see how useful are the movies compared to the fixed snapshots that you can have in if you don't use live cell imaging. Now, going back to the English model, you remember of the simulation of the strand breaks. You clearly see that if you make a Monte Carlo simulation of an electron track coming from gamma rays and a heavy ion track coming from alpha particles, the probability of having complex clustered damage is, of course, much higher when you have high electric radiation. And as I told you last time, can we prove that these clustered damage are important? Actually, one of the best experiments that prove that clustered complex damage are important for the fate of the cell is this one, which is actually quite recent, was published on the Proceedings of National Academy of Sciences USA in 2011 by the group of David Chen at UT Southwestern in Dallas, in Texas. This is actually 3 proteins. One is 53BP1. 53BP1, you have seen already, is a marker of DNA double-stranded break. Then there is XRCC1, and XRCC1 you also have seen is a marker of single-stranded break. And finally, there is HOGG1. HOGG1 is an oxidative base marker. So essentially, it's a marker of a base damage. So when you see, and sometimes you see, this is in the mock irradiated. So this is the control. These are cells irradiated with silicon ions, 1 gray, 10 minutes after irradiation. When you see an overlap of the 3 colors, so let's say of this purple, green, and red, this means that you have a complex clustered damage because you have single-stranded break, double-stranded break, and base damage together in the same region. And this you see happens certain number of times, 39 times here with silicon. You see out of this 11 times is only XRCC1, 5 times is 53BP1 and XRCC1, and so on and so forth. What is really interesting is that if you wait some time, there is of course a repair. This is silicon, this is iron. Iron, it's even higher LET. If you wait, there is repair. And when you look at the residual breaks, so now this is what is left after 24 hours repair, most has been repaired. You see how many there were at 10 minutes, very few at 24 hours, but those that are left are almost all of them are in overlap. Almost all of them are complex damages. Instead, if you use, for example, H2O2, which is also toxic for the cells, you know, it's a hydrogen peroxide, there is never clustered damage if you use hydrogen peroxide. If you use gamma rays, there is something, but not much. Much more when you have heavy ions, showing how the complexity affects the ability of the cell to repair the DNA damage. And what do you do if you cannot repair the DNA damage? You know, what do you do if you are in a situation where the damage is so complex, the DNA is chopped in so many pieces that you cannot deal with it? Well, I told you already, one way at this point is to say, okay, never mind. What we can do, we can use resection. So we can go into alternative non-homologous end joining, cutting the damage and looking for microhomology. This happens, you see, this is G2, in red is G2, and in blue is G1. When you go to X-rays, low LET, also here you see the RPA is a marker of resection. So you see in G1 is the black for X-rays, and G2 is the gray. When you have X-rays here, 2 kV per micron is 250 kilovolt X-rays, some 20% of the cells in G2 undergo resection. Who are these cells? These are the cells that are using homologous recombination. If you use homologous recombination, you see this is actually the marker here is RPA, which is a marker of resection. So if you use homologous recombination, you can use RPA. You don't use always homologous recombination. I told you this already. Not 100% of the breaks in SG2 are processed by homologous recombination, only some 20% with gamma rays, much higher, almost 100% in SG2 when you have very high LET. So this means that even in SG2, the cells resort to the slow, very accurate, but very slow homologous recombination, only if the break is very complex. Now, if you are in G1 though, with X-rays, you cannot use homologous recombination, of course, and you don't use RPA, which makes sense, because you should not use RPA. You should go into canonical non-homologous end joining. But you see, if you increase the LET, then you start to see more and more cells. So you see at 170 kb per micron, you have about 30% of the cells using resection in G1, where resection should not be used unless you want to go into alternative non-homologous end joining. So you see, here is the evidence that alternative non-homologous end joining is LET dependent. And this is somehow, you know, this LET dependence was predicted by physicists many years ago. Well, this is a recent experiment. This is an experiment that we did at GSI, was published in 2016 or 2017, I believe. This instead are old simulations from the year 2000 from the Oxford groups, showing the frequency of double-strand break plus, you remember this classification, double-strand break plus plus with different radiation. These are electrons of different energy, these are protons of different energy, and these are alpha particles of different energy. And you see that for the DSB++, that will be the most difficult to repair, you know, these are clustered damage. When you have slow alpha particles, you start to have a high fraction of them. But with electrons, even when you go to low energy electrons, like 300 electron volt electrons are very low energy electron, the LET is not high enough. You see? And even with protons, the fraction remains very low. And you see, this is pretty much the same thing, but expressed as a plot now, the percent of double-strand breaks, the LET, and you have the, these are DSB plus DSB plus plus, so they are all the complex. These are the DSB plus, and these are DSB plus plus. You see that this is the region of protons, this is the region of alpha particles, and this is the region of electrons. So you see that the complexity of the damage, clearly the simulations show that they increase with LET, and this increase with LET, as we have seen in the modern results from molecular biology, these are simulations from the year 2000. The experiment that I've shown you from David Chen from 2011, 2012, so almost 10 years later, they confirm with molecular biology what was hypothesized with Monte Carlo simulations, that the complexity determines the fate of the double-strand break. Of course, if you go to high LET, if you go to DSB++, what will happen is that you chop the DNA in pieces that are smaller and smaller and smaller. This was known, also this was known since many years. These are old data from gel electrophoresis. This comes from a Swedish group in the 2000s. So very high doses. You see 30 gray, 100 gray, 150 gray. This is the size, and this is the frequency. In principle, what you should have a random distribution. Now, if you have a random distribution, the random distribution should look like that. This should be the random distribution if you cut randomly the DNA. You see that if you take photon, the white points, photon irradiation is actually almost random, you see? It's almost random. After, you have to make a correction for the control. These are the control data. But after correction for the controls, the photon is really random. But you see that high LET radiation is not. This is particle radiation. There is a overexpression of small fragments. So essentially, you chop the DNA in pieces that are smaller. Now, they can be, this is also one big question. There can be pieces so small that the Q will not be able to bind them. And if the Q will not be able to bind, this small piece will actually leave the nucleus, and eventually they will end up in the cytoplasm. So you can leak some DNA because it's simply not bound by the Q if it is small enough. You know, this tail here at very small fragment size. And finally, I just wanted to show you that this behavior of linearity of the induction of double-stranded breaks and repair with an exponential function, essentially, or double exponential function, is true for essentially all phases of the cell cycle. Here you have X-rays in white and iron ions highlighted in black. You see that G1, S, and G2, you always have a linear increase in the number of gamma H2AX, which is actually, this is important, same numbers after iron and gamma rays. So you don't really increase the number of double-stranded break, but you increase the complexity, because you see when you go to repair, the repair with gamma rays is very effective, but the repair with iron ions is not very effective. So again, the impact of radiation quality on the repair of the damage. In the next lecture, we will make one step inside. So let's say we will see how the DNA damage is transformed into chromosomal aberration. So now we will look from the 2 nanometers, we will move up at some 5-10 micrometers. So we look now at the sides of the chromosome to see how the chromosome is impacted by the DNA damage itself.